Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello everyone, this is Joseph again from episode 20 on clinical studies. If we haven't met, I am a PhD student in the Rankin Lab studying at the University of British Columbia, researching the genetics of Parkinson's disease using C. elegans as a model system. In the next couple episodes, I will be going through papers contributing to novel drug development done up to cell models. If you are interested in research using animal models, you should check out our episodes on preclinical testing, episodes 19, 21, and 22. Today, I will be covering papers that provide insight on multi-targeted drug development pertinent to Alzheimer's disease, published in June 2020. We will be going over 12 papers in this episode. Just as a reminder, all of our episodes come with a time-stamped bibliography. If you hear an article that you'd be more interested in, you can check out the paper. To subscribe to this feature, please check out the show notes. Now, let's get started. While these papers aim to target more than one pathological factor in Alzheimer's disease, these first few papers seem to focus a little bit on amyloid beta and tau-mediated toxicity. Check out episodes 13 and 14 for papers delving more into understanding the mechanisms of these pathways. Our first paper is titled Antialzheimer's Disease, Molecular Mechanism of Acori-teterinaui Rhizoma, based on network pharmacology, by first author Li and last author Zhang. This paper is published in Medical Science Monitor Basic Research. Acori-teterinaui rhizoma, or as I refer to it as ATR from now on, is a traditional Chinese herb that is used to treat Alzheimer's disease in Oriental medicine. Here, the authors study the potential mechanisms and molecular targets for Alzheimer's disease by using a combination of computer analyses and in vitro studies. Possible targets of the active ingredients of ATR are predicted by PharmMapper, and the targets related to Alzheimer's disease are filtered by DisGenNet. Protein-protein interaction network of possible targets was then established by a series of analyses such as string, GO enrichment, and KEGG pathway enrichment analyses. Then, the authors used Cytoscape to build component-target interaction networks. Additionally, a gene-to-disease pathway network was constructed and verified by the molecular docking methods. Additionally, mRNA expression levels of APP, tau, and core target genes are estimated via qRT-PCR to study the effects of ATR on amyloid beta 142 mediated toxicity in SH-SY5Y cells. Results from in silico analyses showed that the active components of ATR target Alzheimer's disease-related biological processes such as inflammation, cellular, and metabolism. Some of these predictive targets can be of special interest in future research. Also, the authors found that one of the active components of ATR, beta-caryophyllene, reduced mRNA expression levels of APP, tau, and other Alzheimer's disease-related biomarkers. On to our second paper, Utilization of the OliveNet library to investigate phenolic compounds using molecular modeling studies in the context of Alzheimer's disease, by first author Liang and last author Karagiannis, published in Computational Biology and Chemistry. Adopting a Mediterranean diet has been shown to reduce incidence of Alzheimer's disease. And because of that, phenolic compounds in extra virgin olive oil, a component heavily featured in Mediterranean cuisine, has garnered some attention. To date, a total of 222 phenolic compounds derived from extra virgin olive oil have been studied and characterized in the OliveNet database. In this study, the 222 phenolic compounds, are screened for their ability to bind forms of amyloid beta and tau proteins using molecular docking analyses. A few of these phenolic compounds are found to bind strongly to A beta 1 to 40 and A beta 1 to 42 monomers. In addition to tau fiber protein structures, these compounds are proposed to be novel lead compounds for further research in drug development for Alzheimer's disease. The next paper is titled Dual Targeting of Monomeric Tau and Alpha-Synuclein Aggregation, a new multi-target therapeutic strategy for neurodegeneration, by first author Gaber and last author Peccati. This paper is published in ACS Chemical Neuroscience. For a brief introduction, accumulation of misfolded protein aggregates are a shared hallmark in two of the most common neurodegenerative diseases. Those with Alzheimer's disease have neurofibrillary tangles, and those with Parkinson's disease have Lewy bodies. The precursors to both of these aggregating phenomena, tau protein and alpha-synuclein, have shown synergistic aggregation and neurotoxicity in multiple studies. Here, the authors look to design a novel screening platform for compounds that can target both monomeric tau and alpha-synuclein aggregation to use for developing multi-targeted therapeutics for neurological disease. This new screening platform utilizes cellular fluorescence resonance energy transfer based high-throughput screening for tau-binding compounds. Then, it was followed up with thioflavin T assay and dynamic light scattering measurements to screen for ability to inhibit alpha-synuclein. In developing this new method, the authors identified MG2119 as a promising compound. In an SY5Y neuroblastoma cell model, they showed that MG2119 can rescue combined tau and alpha-synuclein-induced toxicity in a dose-dependent manner, validating their screening platform. Our next paper has a long title, so here we go. 125OH2D3 alleviates amyloid beta 25 to 35 induced tau hyperphosphorylation, excessive reactive oxygen species, and apoptosis through interplay with glial cell line-derived neurotrophic factor signaling in SY5Y cells. This paper was published in the International Journal of Molecular Sciences by first author Lin and last author Lin as well. The active form of vitamin D, or 125OH2D3, and for the purposes of the summary, I will refer to this compound simply as vitamin D, has recently shown potential for treating amyloid beta pathology and is considered a neuroprotective agent. However, the mechanisms driving its effects are not fully understood. Here, authors investigate whether vitamin D ameliorates amyloid beta neurotoxicity by acting on glial cell line-derived neurotrophic factor signaling, or GDNF signaling. In these experiments, SY5Y cells with amyloid beta 25 to 35 are treated with vitamin D with or without a GDNF inhibitor, heparinase 3. In addition to validating previous findings by showing that vitamin D ameliorated amyloid beta-induced reactive oxygen species, apoptosis, and tau protein hyperphosphorylation, the authors also found that these effects were abolished by the presence of heparinase 3, suggesting that neuroprotective effects of vitamin D is dependent on GDNF signaling. In the next few papers, we'll hear a lot about inhibiting cholinesterases in particular. Here is a quick explanation on why cholinesterases are so popular as a drug target. One key feature in the brains of those suffering from Alzheimer's disease is a decreased amount of the neurotransmitter acetylcholine. As a strategy to maximize the already low supply of acetylcholine, drug developers aim to slow down the rate at which it is broken down by cholinesterases. Lo and behold, it works in alleviating many symptoms of Alzheimer's disease. So that is a little insight into why targeting cholinesterases have continued to remain a popular strategy for drug developers. Our fifth paper of this episode is titled Exploration of Dual Fatty Acid Amine Hydrolase and Cholinesterase Inhibitory Potential of some 3-hydroxy-3-phenacetylindole analogs. This two-author paper is published in the German journal Archiv der Pharmazie by first author Tripathi and second author Ayanan. You have to excuse me for my pronunciation here. Without going too off track, once the endocannabinoid system is implicated in Alzheimer's disease pathogenesis, fatty acid amide hydrolase, an integral protein to the endocannabinoid pathway, has been scouted as a target for Alzheimer's disease therapeutics. For the purposes of this episode, I'll refer to fatty acid amide hydrolase as its written acronym FAAH or FA. Inhibiting both FA and cholinesterases have been a novel therapeutic strategy for Alzheimer's disease-related neurodegeneration. Here, authors screen a library of 3-hydroxy-3-phenylsalocindole analogs for FA and CHE inhibition, or cholinesterase inhibition. They identified one such compound with very promising results, showing a balanced multifunctional profile for both FA and cholinesterase inhibition. With structural analyses, the authors determined that the oxidole moiety provided the molecule some rigidness that improved adherence to the compound with target enzymes. Among other predicted findings, the authors also show that compound 16, which is that compound, is non-neurotoxic and can have antioxidant potential. All in all, compound 16 should be further explored for potential multitargeted therapy for treating Alzheimer's disease. Our next paper is titled Comparison of Phytochemicals, Antioxidant and In Vitro Anti-Alzheimer's Properties of 27 Morus spp cultivated in Thailand. This paper is by first author Tambiriyankul and last author Suti Samsame. Excuse me for that one. Published in the journal Molecules. Mulberries, or Morus spp, are shown to have antioxidant activities and have displayed inhibitory activities against key Alzheimer's disease-related enzymes such as acetylcholinesterase, butyrylcholinesterase, and beta-secretase 1. Interestingly, the phytochemical profile of mulberries, even of the same cultivar, vary quite widely depending on the location at which the trees are grown. Here, the authors compare the phytochemicals, antioxidant activities, and inhibitory activities against Alzheimer's-related enzymes in 27 different cultivars of mulberries planted in the same location in Thailand. In addition to validating previous findings that mulberry extracts exhibit neuroprotective and antioxidant properties, they propose one cultivar to be promising for further development as a treatment for Alzheimer's disease. Moving on, in a paper from Drug Design, Development and Therapy by first author Ahmad and last author Mahmoud, we have the title Comparative Cholinesterase, Alpha-Glucosidase Inhibitory, Antioxidant, Molecular Docking, and Kinetic Studies on Potent Succinimide Derivatives. In this study, the authors synthesized two novel derivatives of succinimide and tested them for inhibitory effects against acetylcholinesterase, butyrylcholinesterase, and alpha-glucosidase. They find that both compounds have considerable inhibitory activities, and compound 1 was more potent against all in vitro targets tested compared to compound 2. It also had anti-radical properties. Molecular docking analyses validated the in vitro studies showing a triple-fold acetylcholinesterase and butyrylcholinesterase binding activity for compound 1 compared to compound 2. Our next paper is titled Synthesis of Furocumarin stilbene hybrid as a potential multifunctional drug against multiple biochemical targets associated with Alzheimer's disease by first author Agbo and last author Moore, published in Bioorganic Chemistry. In this paper, the authors synthesized a series of furocumarin stilbene hybrids and studied them in vitro for inhibitory effects against acetylcholinesterase, butyrylcholinesterase, and beta-secretase, and other Alzheimer's disease pathology-related proteins. Among these hybrids, one of them exhibited significant inhibitory activity against multiple Alzheimer's disease pathology-related proteins, including those that I mentioned before, in addition to showing antioxidant activity. Molecular docking analyses of this compound into these proteins were also performed to validate their findings in silico. Next, we have a paper titled PDML Modeling for Alzheimer's Disease, Design and Prediction of Virtual Multitargeted Inhibitors of GSK3B, HDAC1, and HDAC6. This two-author paper is published in Current Topics in Medicinal Chemistry by first author Kleandrova and last author Spec Planche. The advent of computational models of drug discovery have been instrumental for developing therapies for Alzheimer's disease. However, most of the computational models to date focus on a single targeted approach and rely on relatively small data sets of structurally related molecules. Here, the authors developed the first model for simultaneous prediction and design of inhibitors of Alzheimer's disease-related proteins, combining perturbation theory and machine learning based on artificial neural networks. This model, coined the PDML-ANN model, was developed from a dataset retrieved from Campbell, CHEMBL, a manually curated large database of small molecules with drug-like properties, and predicts chemicals to be active or inactive towards the target. Based on results in the model, the authors assembled 10 molecules from key fragments predicted to have positive contributions. Using the same model, these molecules are predicted to be multi-target inhibitors. This work yields promise towards the design of multi-target inhibitors for treatments for Alzheimer's disease. Our next paper is titled Synthesis, Characterization, Molecular Docking, and Biological Activities of Some Natural and Synthetic Urolithin Analogs by first author Gokhan and last author Ghazi. This paper is published in Chemistry and Biodiversity. Urolithins are compounds formed within the gastrointestinal tract following an L-argitanin-rich diet. Foods that are rich in L-argitanin include pomegranate, nuts, and berries. Studies have shown that food rich in L-argitanin can be neuroprotective and thus be a possible alternative therapy for Alzheimer's disease. The authors here believe that the neuroprotective effects of L-argitanin come partially from inducing the production of urolithins in the gastrointestinal tract. In this study, major urolithins and their metabolites are screened for their efficacy in inhibition of various Alzheimer's disease-related enzymes and antioxidant effects. These results point out possible mechanisms of the activities of the urolithins for treating Alzheimer's disease, supported by structural activity relationships and docking studies. Our penultimate paper is titled Insight into Potent Leads for Alzheimer's Disease by Using Several Artificial Intelligence Algorithms by first author He and last author Chen, published in Biomedical Pharmacotherapy. Several proteins have been linked to pathogenesis pathways for Alzheimer's disease and considered potential targets to treating the disease. These proteins include the protein GSNOR, complement factor D, and complement factor 3B. In this study, the authors use a network pharmacology-based screen for potential traditional Chinese medicine candidates that can dock well with these proteins. Artificial intelligence algorithms employing machine learning and deep learning models were used to validate docking results. Their methods predict the herbs Ardisia japonica, Ligosticum chanson, and Lepia nodiflora, and Mirabilis jalapa to contain key components to be a potential effective treatment formula for Alzheimer's disease. Our last author of the episode is a two-author paper titled Plausible Compounds Drawn from Plants as Curative Agents for Neurodegeneration, an in silico approach, by first author Thiruchitapallam and last author Weerasinghe, published in Journal of Computer-Aided Molecular Design. Chelation Therapy has been an approach for drug development to treat neurodegenerative disease. This approach stemmed from the observation of heightened metal ions in patients with these diseases and their implications in pathogenesis. Here, the authors screened for a library of around 200 chemical compounds isolated from Sri Lankan plants for good chelation ability for multiple metal ions. Here, they predict 12 Sri Lankan plant compounds to be good leads for developing treatments for Alzheimer's disease, Parkinson's disease, and prion disease. However, they couldn't do the same for Huntington's disease. Well, that's a wrap for this episode. Thank you for listening, and please do check out our previous episodes covering literature spanning a whole array of topics related to Alzheimer's disease. If you don't know where to start, might I suggest episode 20, a short episode where I covered papers about clinical studies. We're always looking to hear from you, and feel free to reach out to us on social media. That said, over to Sarah for some final words. Goodbye, and see you next episode.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout. We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.